I have the pleasure to announce the next panel, and the next speaker could be said to be the father of spirit and opportunity, Phoenix and curiosity, besides Mars Reconnaissance Orbiter and Maven, as he, as NASA's chief scientist, wrote the book on the scientific strategy that led to these missions. It is safe to say that James Garvin is my favorite chief scientist and has been for almost 2 decades. James, please come on up and introduce us to your extraordinary science panel on Mars.
Well, thanks, Artemis, and we're going to switch the nameplate so no one gets confused. We're all Martians here now, so I'm Jim Garvin, and thanks, Artemis. Now for something completely different comes to mind. We'd like to regale you with Science at the Frontier with 3 extraordinary Martian scientists to try to put all this in perspective of what we are learning about this place. So let me bring out, oh, they're coming the other way, sorry. The 3 panelists that will provide content on what it is we're learning about Mars in our quest for answers to issues such as, are we alone? Danny Glavin, and I'll introduce them individually in a second, Jen Eigenbrode and Pan Conrad. John Grunsfeld really said it best. Today, people on Earth, like these 3 colleagues, are exploring the real Mars, really in the spirit of a scientific revolution of understanding this planet. But also, these are the kind of colleagues that I would submit to you we'd like to be on Mars today, or soon, maybe not today. And that's really why we've invited them to provide content for this session. So let me just remind you that Mars is a big place, and as we talk about going there with our robotic explorers and with people, when we look at a place with literally 150 million square kilometers of real estate to explore, the question comes to mind, how do we ask sensibly or address the issue of, are we alone? And so today's panel, through the lens of what we're learning today and what we hope to learn in the next decade, are going to address that in 3 ways. So let me introduce them. First, Dr. Daniel Glavin. He's the Associate Chief for the Solar System Division at NASA Goddard Space Flight Center, a member of the Curiosity Science Team. He's the author of seminal research in understanding amino acids from space materials such as meteorites. And he's going to address the challenging question of how should science pursue the ideas central to the search for extant life on Mars now and across the next few years. Next, from my left and your right, or whatever, I got it backwards. Okay, it's okay, sorry about that. It's hockey, you know. The next colleague is Dr. Jen Eigenbrode, and really Jen speaks for the future of Mars exploration science, both here on Earth and on the surface of Mars. Her work is really linking the record of biology in rocks, in a rock record, both here on Earth and we hope on Mars, to that which we can actually explore on Mars now with Curiosity in her role as a science team member on the SAM team, but in the future as we move forward. And Jen, while looking for water on Mars, also enjoys kayaking on water on Earth. Love her to kayak on Mars. I'm not sure how, but physics is always willing. And finally, to my right and your left is Dr. Pan Conrad, and Pan really goes without words. She is the Deputy Principal Investigator on the Sample Analysis at Mars investigation, which is central to Curiosity, our mission developed by JPL that's been on Mars 975 days, I think, today. Maybe off by a day, sorry. And Pan really, aside from being a brilliant musician, which I didn't fully appreciate, Pan, sorry about that, is also, so I think music carries forward astrobiology, is also one of our leading astrobiologists. So today, Pan will talk about sort of the systems view of how we explore this big Mars scientifically and get ready for what's going to come in the future. So the format of the panel, very simply, will be each speaker will speak for approximately 10 minutes, going through their content, raising questions. They will go in order, Danny, Jen, and Pan. Afterwards, I will address one general question to the panel and then open it up to all of you. This is Science at the Frontier, so we hope we will at least provoke some interesting questions. So let me turn first to Danny. If you'd like to come to the podium, I'm going to hand him the magical device to move the slides. Thanks, Danny.
Okay, thank you, Jim. These are really exciting times for Mars science, as we've heard from Grunfeld and others. Curiosity is really just giving us some amazing results. Is this clicker? Okay, here we go.
There you go.
Oh, I'm not Garvin.
Oh, sorry, Danny.
That's okay. So before I begin any speech about the search for life and the strategy, it's always good to kind of remind everybody what the basic requirements for life as we know it here on Earth, and also to remind you that this search is highly biased. We only have one example of life, and that's life here on Earth, terrestrial life. So all of our requirements, what we think a habitable environment would entail, are based on this. We need the basic major biogenic elements: carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus. Energy sources, some very obvious, like sunlight, but others not so obvious, like minerals, for example, in the rock. Oxidize and reduce minerals that could provide a redox gradient for chemical energy that could be harnessed by bacteria. And then, of course, there's a major reason why NASA the Mars exploration program has chosen to follow the water, and that's the fundamental requirement of liquid water and all life on Earth. And we've nailed it on Mars. I mean, the Curiosity rover has found an environment in Yellowknife Bay that has all of these in the past, ancient mudstone. Grunsfeld also mentioned the really exciting discovery of complex organic material. I mean, this is fascinating. I mean, in my wildest dreams, I was very skeptical that we'd find it because of the ionizing radiation and oxidants that can destroy organics, but there are organics preserved on Mars. We're ready for that next step, trying to figure out if this environment ever supported life. And there are a variety of ways to do this. What I'm going to focus on in this talk is primarily the search for chemical biosignatures, the building blocks of life, compounds that can self-replicate like DNA and RNA, amino acids that possess chirality, a unique property of all life on Earth, and then, of course, carboxylic acids, fatty acids, and lipids that make up cell membranes. These are the compounds we want to go after. Is Mars inhabited today? Well, this is the question that we're all asking. From the looks of the surface, it's not a very hospitable environment, right? Extremely cold, very dry, low pressure, about 1/100th the pressure on Earth's atmosphere. Liquid water is not stable on the surface for long periods of time. Only remnant magnetic fields, so you don't have the magnetic fields on Earth to shield you from the harmful ionizing radiation that can destroy organics. There's no ozone layer, no thick ozone layer on Mars, like we have on Earth, that can shield you from UV radiation. So these are all challenges we face. Nevertheless, there is an environment in the subsurface that isn't exposed to all these dangers, where you could have higher pressures. Liquid water could be stable environments that could support life. So I'm actually pretty optimistic that our search will take us to these places in the future. I'd be remiss if I didn't mention the Viking missions to Mars. To date, the only experiment geared towards detecting extant biology. These were the gas exchange, the labeled and pyrolytic release experiments on the Viking landers. A lot of attention has been drawn to these experiments lately, in part because the labeled release experiment actually gave a positive result. 14 carbon-labeled CO2 was released from the soil when carbon-14 spiked nutrients, organics, were added to the soil. This was really intriguing, and it wasn't found after the soil had been heated. What we now know from Mars is that there's a powerful oxidant in the soil, okay, that can react with organics to release CO2. So the general consensus is that these results were inconclusive for life, since there's a non-biological explanation, and that is these chemical oxidizing processes in the soil that can provide these results. I have to say that after working on the sample analysis at Mars Instrument for the past 10 years now, I have a new respect for how difficult it is to not only build a flight instrument and land it on Mars, but to design it in a way that can be responsive to all the unknowns that you face in any environment. And Viking was the first to do this, and obviously didn't know a lot about the chemistry of the samples that it was analyzing. We heard about methane on Mars. This is a huge discovery. Essentially, this means Mars is not the dead planet we thought. It's an active environment. Whether you think this methane is from biology, methanogens in the deep subsurface, or from geology, it doesn't matter. Methane is not a gas that's stable in the atmosphere for long periods of time, has a lifetime about 300 years, I think. And so the fact that we see it, and we see it in plumes, means that there has to be an active source. Curiosity, as it was mentioned, recently confirmed this from the ground in Gale Crater, but methane's also been detected from Mars orbit as well as from ground-based telescopes on Earth. So Mars is an active environment. This is exciting. Now, where should we look for extant life on Mars? Well, go back to the requirements for life. You want an environment that has organic compounds, nutrients, an energy source, and liquid water. The surface may not be that place. So ExoMars rover has a subsurface drill that can go down to 2 meters to get below this sort of ionizing radiation damage zone and potentially access ice or even potentially liquid water to look for evidence of life. Folks have talked about South Polar ice cap drilling missions to actually look for organics in the ice cores themselves. And even interesting, we know that there is evidence of liquid water, at least for short periods. We see this from orbit and these RSLs or gullies. So you get these saline fluids that kind of come out from the side of craters onto the surface. Perhaps these environments might be good places to look for extant biology. Now, I will point out that one real challenge associated with going to these, I'll call special regions, okay, regions where you could have ice or water within a meter of the surface, is you have to sterilize your spacecraft. We know how to do that. Viking did it, so it's not a technological challenge, but it is, obviously an extra cost to the mission to do this. And this is all because of the planetary protection requirements that dictate that we shall not contaminate Mars with terrestrial organisms that could potentially grow. So what should we look for? And again, I'll go back to these chemical biosignatures that I pointed out in the first chart. Amino acids and nucleobases, we know that these are fundamental components of all life on Earth, and proteins and enzymes. The nucleobases are genetic code, essentially, in DNA and RNA. What's really cool is that we see these compounds in meteorites. In fact, hundreds of amino acids have been found in carbon-rich meteorites, like the Murchison meteorite, and even some components of our genetic code in DNA. The nucleobases have been discovered, and these are extraterrestrial in origin. So these compounds are being formed in space. They can be delivered to the early Earth, contributing to what we call the prebiotic soup for the origin of life, but they're also going to be delivering this organic material to Mars. So there's no reason why early Mars couldn't have had a prebiotic organic soup from which life could emerge. One of the really interesting things and big mysteries in origin of life research is why life has only selected a limited set of these compounds. Despite there being hundreds of these compounds available in the prebiotic soup, we only have about 20 amino acids in our proteins and 5 nucleobases in DNA and RNA. Perhaps this is the type of signature that we should be looking for on Mars. We look for these compounds, but more importantly, we look for a limited distribution of these compounds. If we see hundreds of different compounds of every possible structure, perhaps that's an indication of non-biological chemistry. We can look for unique distributions and perhaps even chirality or handedness. All life on Earth is based on left-handed amino acids and right-handed sugars. I've always said that one of the coolest and most astounding discoveries for me, from an origin of life perspective, would be to detect a limited set of these compounds on Mars, but more importantly, a completely different set of the 20 amino acids, and perhaps the opposite chirality, so right-handed amino acids and left-handed sugars. That would provide definitive evidence of an independent origin of life. I'll say that again, an independent origin of life. Why is that important? Because we have no idea how difficult it is to go from a prebiotic, non-biological world to a biological one. We have one example of it. It happened here on Earth. Did it happen elsewhere? If we could show that it happened twice in the same solar system, wow, there should be life teeming everywhere. Probability just went up. So return samples from Mars by robotic and human missions will certainly help in this search to try to answer these questions. Is life, did it only originate once here in our solar system on Earth? Are we really alone, or did this happen elsewhere? One of the really cool things about the human missions is they'll be able to provide a wide variety of rocks that are specially selected, and frankly, just a lot more material than we could get back with robotic missions alone. So really looking forward to seeing that happen and hopefully answering some of these really fundamental questions. And with that, I'll pass it on to Jen. Thanks.
I'd like to take a few steps backwards. Let's talk about why we are going to Mars in the first place, to look for evidence of life. If we think about early Earth, we had water, we had lots of energy from the solar radiation coming down, we had impact events, there were volcanic eruptions, all sorts of things were going on. There was heat sources, there were nutrients everywhere, and somehow, some way, life took hold. Now, elsewhere in our solar system, that may have happened on other planets. Mars happens to be our close neighbor, and we have suggestions that tell us that it might have been much the same way. Water, energy sources, volcanism, impact events, nutrients abounding, everything is in motion. Could life have started on Mars too? For this reason, there is a very fundamental basis for why we're interested in going to Mars. If life took hold on Earth during early Earth's history, perhaps life took hold on Mars too. All right, now let's go to today. Things have changed quite a bit, right? Earth has evolved. I mean, we have an amazing biosystem with incredible diversity. It's all over the place. You cannot go anywhere, look any place, and not find life. It's just ubiquitous across the planet. On Mars, Mars took a very different path. It's this seemingly stale, boring red planet sticking out there. Of course, our perceptions are changing. But what's really astounding is look at the difference between the two. They took very different paths. If life started on Earth and Mars in their early histories, then what happened to life on Mars? Where did it go? Did it evolve as well, just like life evolved on Earth? Where would we find the evidence for it? In this type of pursuit, we still have to take a couple steps towards where we are here, our home, and take the lessons from what we have learned on Earth. And the fundamental one that I think that is just, it's made a huge difference in my career in the science that I've done, and I think that it has affected all of our astrobiology endeavors. And that is, life exists in all the extremes on Earth. It's every time we think, no, there's no way life could live here, we go and we find out, yeah, it sure is there. It's doing something. Let's go through a few examples. The Atacama Desert up here in the top left. Atacama Desert is an incredibly dry place. When it rains every 10, 15 years, and when it does, you'll find a few little raindrops on the ground, and that's it. It's like there's next to no water there. It's so dry that we have a hard time finding evidence of life there. It is there. It's not doing much, but it's hard to find it. So when we go to the Atacama, it feels like you're walking on modern-day Mars. It's so dry, you can't find any visual evidence of life. You actually have to dig into the sediments and extract organic molecules and cells from the sediments in order to find it. Now, if we look at the top right side here, this is Dalalo, Ethiopia. It is so acidic, the pH is less than 1. It's also hot, very hot. These are hot, acidic springs. I mean, it's like living in hot battery acid, and yet organisms do. In fact, there's a lot of them. You look at all these different colors up there, those different colors, a lot of those are from pigments from the organisms. Life still lives there. Let's look at the bottom one on the left side. That is Blood Falls down in Antarctica. Blood Falls is this rusty iron water coming out of a lake that's underneath the glacier. It has no light source. It is limited in all of the inputs that go into it. It is a secluded environment. And when the water comes out, this is what we see. And yet life is still there. Life has found a way. Now, the most amazing one, in my opinion, and the closest one that I think relates to Mars today, is Chernobyl. We're all familiar with Chernobyl. Major amounts of radiation all over the place. And yet, they have found fungi living in that radiation environment. They're not just living there, making their way through things. No, they are thriving on the radiation itself. They actually grow using that radiation. Somehow they have found a way to do that. In all of these instances, they teach us that life adapts to their environment. They find a way to make themselves do what they need to do to survive. They don't always do it very fast, but they do. So, let's assume that life has existed on Mars in the past, and maybe it's found a way of adapting to all of the environments that Mars has experienced through time. What ancient biosignatures might we actually look for? Well, just like we have a reptile that might die, end up in the sediments, and we uncover a fossil, we have other types of fossils that end up in the rock record from microorganisms. For instance, if you have a microbial cell, the cell wall can give you a bunch of lipids. Now, we're all familiar with fatty acids. We're told that we need in our diet and everything, or fish fatty acids and whatnot. You know, all of those fatty acids feed our system, and a lot of them end up in our cell walls. And those cell walls aren't just a whole bunch of fatty acids, like what we would find in a meteorite. We do find them in meteorites. No, life is selective at what type of molecules it uses. Even though it may have all of this diversity available to it, it says, I want this one, I want this one, I want this one, and I want that one. I don't want the rest of them. Or it only synthesizes those set. Life is selective. So even though we have very specific molecules that we look for on Earth to understand if there's evidence of life in the past, what we can gain from that knowledge is that life is selective, and we would expect on Mars, even if they didn't live the same way they do here on Earth, that life would still have been selective. You won't get all of the molecules like you would in a meteorite from non-biological processes. Instead, you would get a selective set of molecules. And that's what we would look for. That type of information tells you about biosynthesis. It gives you first insights into metabolisms. And that type of information is incredibly powerful in understanding an ecosystem. What else might we look for? Microfossils. If we have the right type of microscopes, we might be able to uncover those. Every biologist that I have talked to who studies extreme environments says they want to see a cell. Where it's possible. They are in the fossil record. If we have microorganisms living in a wet place, they might make a mat. This is very common on Earth. We call them microbial mats. Those develop textures that might get preserved. And here's an example of some of them. You see these little round circles? Yeah, that's a very specific type of feature that we would relate to microbial mats. These are just a few examples. But these are the types of things we look for in trying to understand what life was like in an ancient environment on Earth. We should look for this type of stuff on Mars. And for all the same reasons that Danny pointed out, you have to have the essential things. You got to have water source that has to stick around a while. You got to have energy source, and you got to have nutrients. For all those types of things, we are looking for those types of places in the Martian rock record. And once we find those, we look for this type of stuff, all these biosignatures. There's another one. This is a bigger picture, and we do this on Earth. We know that on Earth, the environments have changed. On Mars, the same thing would have happened. Organisms, they use their environment, and the environment responds, and the two interact. They're constantly changing. They become one. That's called an ecosystem. The dynamic keeps going and keeps going. So if the organisms start using something because they're metabolizing something, and the environment is losing that, it might change, and the two kind of evolve together, and everything dies, ends up in the rock record, and you end up with a stack of rocks like this. But if you go through and you look at the chemistry of each of these layers, you'll see that it's changing. And those changes in the chemistry and the sediments tell us about how the ecosystem evolved. This is the group of organisms, not just one. This is an incredibly powerful approach to understanding the history of life on Earth, and it would be the same on Mars. And it might tell us why we might have started with something that looked like early Earth and ended up as this red planet. Now, I talk about the red planet being kind of stale and not much happening because when I started this job, that's really kind of where we were. Our perceptions have completely changed. It is amazing to think that, 10 years ago, we had just figured out that, hey, there's water on Mars, and now we know it wasn't just there, it was sticking around. It was doing things. It was changing the surface of the planet. Hey, there were places life could live here. We have figured that out. But what's most amazing is look at this drill hole here. It's just one example. Curiosity rover, which is on Mars right now, drilled that hole. They went through the red crusty layer that we see from here as the red planet. And lo and behold, right below the surface is this dark gray sediment. Oh my gosh. Mars is not just rust. That's just a little bit, that's a layer on the outside. There's a treasure underneath. That rusty layer on the outside is what happens when you have all of these oxidants on the surface and all this radiation bombarding on the surface, and it's changing the chemistry, it's changing what is there. But it might not go all so deep. So what if we go a little bit deeper? Curiosity is going down about 5 centimeters. What if we went down 1 meter? What if we went down 2? All of that chemistry, all of that diagenesis, all of those changes that are happening at the surface because of radiation, because of oxidants that are destroying the record we are trying to get to, might actually have not been altered. Deep down, we might actually find something closer to what we find on Earth, something that hasn't been broken down, the original stuff of an ancient ecosystem. So I propose this idea of what if we could go 1 to 2 meters? What if we could go much deeper than that? Then what? Then what could we find out? How would our perceptions of Mars actually change? I imagine that someday when we have humans at Mars, that we're going to get cores of rock. We're going to drill down, and we're going to pull out these long cores. We're going to look at them. Then we're going to send all of our instruments in, and they're going to look at them. They're going to map it. They're going to tell us what the chemistry is, and we're going to have all of this information, just like we do on Earth all the time. And then that information is going to get fed to an astronaut who's going to turn around and say, that's the sample we want. That's where we want to look for evidence of life. And they may have a robot actually doing the sampling. They may be the ones doing the sampling. I don't know. But the point is that by having the humans right there, there's a cognitive response to the observations that happens on a very fast time scale. And you don't have to wait for all of this information to go back to Earth and come back. I mean, we probably would still do that to some extent, but the point is that you would have somebody right there who could then be responsive to the observations. Maybe they want to fine-tune the observation so that we get the best information we can and do something with it. We want to do something with it. We are looking for evidence of life. And this, honestly, could be ancient life that we're looking for or modern life. Well, we don't know. But in either way, this is a pretty exciting approach. So on that note, I'm going to pass it over to Pan.
Thank you, my dear. Okay, so what I'm going to do is talk about where we are and where I hope we will be. In 1957, my father took me out in the backyard, yes, I'm that old, and we looked up at Sputnik, and he said, someday you'll go up there. In 20 years, I'm going to be pretty damned old. Don't let me down. It takes not only a lot of people to support a mission to build a mission, and to actually debrief the mission. It takes a lot of people to pay for the mission. What I want to make sure I convey today, if nothing else, is that NASA isn't a bunch of suits, even though I do work for NASA and I did try to clean up for you. You are NASA also. You pay our salary, and you do get a voice. And when we send missions to Mars, you all get to participate in the landing site selection. It's open public workshops. And I beg you, please be involved. If you like what you see, come and participate. If you don't like what you see, come and participate. It's a big solar system, and it is gonna take all of us. And what I want to leave you with today will be my reasons for why I think it takes a village on Mars to make the next step. These 3 pictures are all places on Earth. They're all places I have been to trying to find a place where there is no life. I haven't succeeded. There's only less life. The picture on the left is from an Arctic desert. It's called Buckfjord. I'll be back there in about 10 weeks. The picture on the upper right is called Badwater Basin. It's in Death Valley, and that white stuff is what we call precipitate minerals. The stuff on the bottom, lower right, that's in a polar desert. In the other direction, that's Battleship Promontory in Antarctica. In all of these places, life thrives. Not a huge diversity of life, but life is able to exist. And the reason we study these places on Earth is so that we can begin to understand what some of the strategies life would employ if it were to be present on Mars. I'll tell you a little bit more about why I think we need to understand that shortly. But what I want to show you here is a contrast. These 2 places are such that one of them I can get to and the other one I can't. The reason I can't get to the one on the top is because it's on Mars. The one on the bottom is also in the Arctic on Earth, and it's full of life. If you walk up to that outcrop, you'll see organisms living in the rock. This is important. Hold on to the living in the rock thing. If we could get to strata exposed in extreme environments, cliff sides on Mars, we might be able to dig in and see ancient evidence of life. It's very expensive, and it's very risky to do that. When you entrust us with an expensive asset on the surface of Mars, we have to be good stewards of it, so we don't dangle things off of the side of a cliff yet. So this is Mars. This is where I go to work every day. I can't believe that I get to wake up every day and look at these images from my field camp. So my training as a geologist at GW has come in very handy. Look at this. It's not a good site. It's very inhospitable. I personally don't think there's any life there. So here's the question. Is there life somewhere there? Could there be life in refugium on Mars? The reason why I'm giving you this bleak picture of sitting right out on the surface is because it's not just about chemistry. Although chemistry plays a big part, it's also about physics. Physics constrains the kind of chemistry you can conduct. And if nothing else, life is about the commerce of chemistry. Metabolism is chemistry, and you've got to have the right conditions to make it go. The right conditions, as both Jen and Danny have articulated, could exist beneath the surface. If at some point Mars was hospitable for life on its surface, life had to go somewhere else, because sitting right out on the surface right now is not only strong doses of ionizing radiation, which has been measured continuously by the RAD instrument on the Curiosity rover since we landed in 2012, but also extreme chemical oxidation. There's all kinds of chlorine species on the surface of Mars. I like to think of it as a Clorox planet. So you're challenged both by chemistry and by physics. Go down, young man. I believe that it is possible that if we could get beneath the surface, we might find refugium environments. So the way we start is we look at these amazing images, this one taken by the mast cam on the Curiosity rover, and we ask ourselves, okay, what might give us an indication that there is a shallow cave or some protected environment just beneath the surface? If we look at these hills going up to Mount Sharp, or Aeolus Mons as it's formerly called, where we're heading with our rover, we look at this physiognomy, this geomorphology, and we ask ourselves, where would we go to look for a protected environment? The problem with doing this is that we are constrained to cycles of communication and the technology we have, not to mention the human assets who get very tired after a 10.5-hour tactical cycle. So there are real technical, financial, safety, and all kinds of constraints that only allow us to have a small window of science opportunity each day with this rover. Given the fact that we then have to communicate with Earth, then we have to deliberate, then we have to make a decision, then we have to program the rover, and we've got to get that uplink command up to that rover while we have the satellite window. It's a challenge. So one way of addressing this challenge would be to develop a field campaign on the surface of Mars. I don't have to sell all of you. You wouldn't be here if you didn't agree already. So let's talk about how we would do this. I like to cook without recipes, but you can't cook without a recipe on another planet because of all the constraints I've already mentioned. Here's how we do it on Earth. Everything that you see on this list, and I'm not going to read this, you can all read, everything on this list except that last one, randomly select locations and proceed, we can do on Mars. Why can't we do that last one? Well, it's because it's just too expensive and requires too much planning, and there are technical constraints that tell us why we can't just randomly go to any place we want. But once we select a landing site and find what we hope will be a promising location, what we then do is constrain our search space so that we can get the most science for our effort. Exploration without science is just adventure. There's no return. Exploration is great, but the tools that we have at our disposal to bring back societal benefit to all of us, those tools are vested by science. So when we go to the field, we look at the area, we say, okay, how shall we constrain our search space? This is an area in the Mojave Desert. I drew that square there to say, hey, look at this, there's a little bush there. Why is that bush there and not somewhere else? If you look in the background, you'll see a lot of scrub terrain. Well, the truth is this is a floodplain. Ancient, periodically floods when you get an inundation in the rainy season in California, but not very often these days. And occasionally, you get scrub. So the question is, how do we divide up the terrain so that we can get a manageable grid? I drew a square randomly. I found a piece of life in it. And then we marked off smaller squares within there. And about every third of a meter or so, we took samples of that sediment on the top. We dug down and took stuff beneath. The place is full of microbes. We even found a few odds and ends, insects, worms, etc. The point being that what you see on the surface, A, does not represent what you see beneath the surface, and B, you have to systematically explore. The way we can systematically explore with a rover is very constrained. Would we send humans to Mars and not bring a team of robotic assets? Never. My former student, Julia, is sitting there using a reflectance spectrometer in 3 bandwidths to look at how the materials there inform us of their unique nature by the light they absorb and the light they reflect. We would never go to the field with at least some tools, and we're not going to do that on Mars either. I think you understand that it's a marriage between robotics and humans. I want to also emphasize the importance of imagery. We use imagery on Earth. When we go to the field, we're going to use imagery on Mars. The important questions that we ask, we start with imagery all the time. Here's a good example. I told you that there were rocks in Antarctica and rocks in the Arctic and rocks in Death Valley where organisms take up residence. You can't just take up residence in any rock. Why? A, you have to have the right kind of chemistry in the rock, and B, you have to have the right physical properties. This porous sandstone you see on the left side of the screen, which is from Battleship Promontory in Antarctica, is ideal for hosting what we call cryptoendolithic organisms, those that live inside rock. Why? Because the pore spaces that you see on the right allow retention of fluid and mobility of the poo so that you don't drown in your own waste products. So this is a very, very quick tour of some of the things that I think we'll be doing with a human team. I'm working right now with some of you who I have seen out here to develop some human science objectives, and in fact, I'm probably supposed to be at a telecon right now that I'm not on to discuss this, but this is kind of fun. Every time I look at this rover, and I love these selfies that we take, I am thrilled to look at that rover, and I am thrilled to see hardware that I designed sitting on the front of it, hardware that I made, thrilled to see the instrument that Jen and Danny and I use routinely for our experiments sitting inside the belly of that rover. But I look at this outcrop and I say, if I were there, I would be doing this, 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 and this pace is just way too slow for me. So I'm going to stop talking so that you can ask questions and Jim can ask questions, and we'll see where it goes.
Thanks, Pan. So well, let me stand back. So folks, I think from Danny, Jen, and Pan, you got an impression of what it is we're doing and what we'd like to do on Mars. And when we look at a picture like you can see here, taking about 100 sols a day, not a day, on Mars, it sort of gives you a lot of questions you'd ask that I think derive from what you heard. So I'd like to ask our panelists for a very quick amount of time to just comment on really the tough question. It's right above. So, a huge planet, lots of action everywhere, underground, underfoot, under wheel, under whatever. Why don't we just leap in, as we did with Viking? I was a Viking intern, truth be told, so it dates me. Leap in and let's find the living stuff now. Why aren't we just rushing to that judgment? Because that's fun. It's exciting. You guys just said it's everywhere here. So I don't know who wants to begin. Anyone have a thing? I'd like our panelists to comment on that, and then we maybe have time for a quick question. Anyone? Danny.
I guess I can start. I mean, I'll just quickly say we certainly have come a long way since Viking. I mean, but for the fantastic instrument payload it had, which it really did for the time, it was amazing, it couldn't move. It was stuck in that soil prison, I'll call it. Imagine if Curiosity landed and couldn't rove, and we couldn't get to Yellowknife Bay. We couldn't find the habitable environment. Mobility is huge. So I think that gives us a much better chance in the search, coupled with drilling. Right now we're drilling down a few centimeters, but future missions are looking to a meter or more with mobility. Again, looking ahead, I think we're ready for this. I mean, it's not without risk, no question. Mm-hmm. You can be fooled. But I think we're ready to take the next step. I mean, how many times do we have to show Mars as a habitable environment before we actually try to figure out if it was ever inhabited? I'm ready. I think the public's ready. But we need to be smart about where we go, how we sample, and the measurements we use to look at the sample.
Thanks, Danny. Jen or Pan, comments?
Sure. I'm not so sure that we're ready today. I think that we could prepare ourselves to be ready, but there are so many unknowns in the picture. I mean, it's not like science here on Earth where you have this amazing amount of familiarity with what you're working with that we take for granted. There really is a fine line between science research and exploration when it comes to being on another planet, because you're dealing with so many unknowns. You really step in and hope that you learn something that pushes you in the direction you really want to go. And right now, with each mission, with Curiosity, we are learning so much about the chemistry of that planet. If we hadn't that information in our pocket now, we might be sending instruments that might not work because they would react, the perchlorates in the sediments might react with the instrument itself. We have to optimize our instruments based on the knowledge we have about Mars so that we can get information that is at least useful. And I'm not so sure that we're there yet, but I have high confidence that we're going to get there. And like I said, the information we're getting from Curiosity is feeding into that. And you know, the instruments are going to get smaller. We'll have lots of them. Mm-hmm. And I think that we will have learned a lot more about the big picture and the details of Martian habitability that go beyond just checking off those boxes. And we might actually stand a pretty good chance of succeeding.
I'm going to take a contrarian view.
Okay.
We absolutely are there, come on. I think we are ready to look for life, but I don't think it's the right question to ask. So the reason one might want to ask that question if you were getting ready to send humans is to understand whether or not there was something there that would harm the humans. You just heard me tell you the surface is full of Clorox, the surface is full of radiation. On Earth, when we want to kill microbes in a hospital or in a lab, what do we use? So the fact is, I think the surface of Mars is likely to be safe for humans. I think we can do the job much better if we're there than if we send a robotic asset, because some of the things that we need to do, we can't get the robot to do without too much risk to the robot. When you have human in the loop in real time without the latency of the communication between Mars and Earth, you can respond and take bigger technical risks knowing that you can pull the plug should you need to do it. So if we want to ask very complex questions like, is there life here? What are the resources here? I think we need to have humans on the ground there as a team working with the team of humans on the Earth.
Well, I think with that we have to end, but maybe these are the people that need to go. I'm kind of sending my vote. And I just wanted to finish with the one thought that, there we come, whoop, went too fast. That I think what we heard today from our science panelists and from the community, and some are in the room, thank you folks, is we're ready for the challenge. However we go about it, obviously being there, you know, it's like the movie being there. It's going to be great. And really, it's an amazing time for Exploring Mars. So I wanted to thank Danny, Jen, and Pan again for their comments. And over to you. Oh, over to Artemis. Okay, well, thank you, panel. I think we're excused politely. Whoop. No. Thanks again.